The CRISPR, this segment of DNA of this A, B, A, C, A, D, this is some type of bacterial immune system.
Because this is crazy. We're playing God at this point.
So we're actually going to start back in 1987. It was Yoshizumi Ishino. He discovered this segment of DNA in E. coli that had a very weird structure, okay? What it had was, it had these repeating sequences, okay? And in between these repeating sequences were non-repeating unique sequences, okay? So it was kind of like, there was like A, which is the repeating, and there would be A, B, and then A, C, and then A, D, and then A, E, and so on and so forth, okay? It was very weird.
Yes.
Highly structured.
Right.
The A was always the same.
Yes.
Okay? And at this point, this is very new, like, research. And whenever you find this kind of nice mathematical repeating structure, it always like raises flags.
Right.
Right? So this guy, he publishes a paper in 1987 where he's like, I find these consistently repeating segments and then with non-repeating segments. And everyone started focusing on the repeating segment because that's the part that gets highest signal.
Right.
So the repeating part is kind of crazy because it's also a palindrome. You know what a palindrome is?
Yes.
It's like a race car.
Yes.
Race car is a palindrome because you read it front or from the back. It's the same thing.
Way. Yep.
Well, DNA is double-stranded.
Yes.
So there's a top segment, for example, and then there's a bottom segment.
Yes.
And the top segment you'll read from left to right.
Yes.
That's what they call from the 3' to the 5'. And that'll be some sequence of ACTGs. But if you read the bottom segment from the other way, it'll be the same exact thing. So it's already like, not only is it repeating and has this highly conserved structure,
Yes.
but it's also within the repeats, there's this structure.
It's this palindromatic structure.
Yeah. People thought it was interesting, but didn't really think anything of it. Then came Francisco Mojica. This is back when bioinformatics was just a nascent field, and people had started to sequence not just E. coli, but they were starting to sequence viruses. Specifically, these things called bacteriophages. They're viruses that attack bacteria, hence bacteriophage. Okay? And what they noticed was the stuff that people weren't thinking about in CRISPR, which are, you've got the repeats, the A, but you also have these B, C, D,
Right.
which has low signal.
Right.
Those interspaced
Yes.
unique parts were exactly the same DNA as the stuff that we were finding in bacteriophages. Okay? So now it's like, and he comes up with this really cool hypothesis, which is